So we're talking again about cellular division, but in this case we're talking about a process called meiosis, where multicellular organisms create their sex cells. So meiosis, like mitosis, is a cellular division process, but it doesn't happen in the somatic cells of the organism. It only happens in the germ cells. And in this case, germ cells are diploid cells that live in the gonads of the organism. So diploid cells have 46 chromosomes, remember. And these germ cells, through the process of meiosis, turn into gametes with 23 chromosomes. And these gametes are sex cells. So in humans, these are sperm and egg cells. So meiosis produces sperm and egg cells. And this happens with two division processes, meiosis 1 and meiosis 2. So here's the process of meiosis. DNA replication happens, just like before mitosis. And then the process begins with prophase, and then metaphase, anaphase, and telophase. And then there's a division process, and there's another division. So then it's meiosis 2, and you have prophase 2, metaphase 2, anaphase 2, telophase 2, and another cytokinesis. And in meiosis, you get 4 haploid cells in the end. So these are your sex cells. Up here, you'd start with a gamete, a germ cell. And through this process, you create gametes with 23 chromosomes. And this process is interesting because in meiosis, you don't separate the sister chromatids right away. You first separate the homologous chromosomes. So remember, homologous chromosomes are chromosomes that hang out in your body, and you have one from mom and one from dad. When they're duplicated, you have 2 of each. And so if these are your homologous chromosomes, starting with meiosis, they separate here during meiosis 1. Homologous chromosomes separate. And then during meiosis 2, that's where sister chromatids separate. Like they do in mitosis. So there's 2 divisions, and in meiosis 2, sister chromatids separate. This is a pared down version of the 2 division processes. You start with homologous chromosomes. These separate during meiosis 1 here. Homologs separate. And then you can see here, sister chromatids are separating. So those are the 2 division processes. and what they result in. The results of these two division processes are 4 haploid cells. So remember I said you start with your germ cell to 4 haploid gametes. And these gametes are ready for fertilization. The reason why you go from 46 to 23 chromosomes is because when through fertilization, you have to combine 2 sets of chromosomes. And then in the end, you'll have a whole organism. But if you didn't first go through mitosis, you'd have way too many chromosomes in your progeny. So something else that happens during meiosis is genetic recombination. So we're paring our DNA down into 23 chromosomes, and we also are creating genetic variation. So through meiosis, new combinations of genes arise. And this is how sexually reproducing organisms create variation in their DNA. So through these mechanisms of variation in meiosis, you come out with a new generation that's different from the first one. So one of the mechanisms of variation in meiosis is called crossing over. And this happens during prophase 1 of meiosis. Remember, there's 2 division processes. So in the first division process, in prophase 1, these homologs, your homologous chromosomes, get together. So they find each other, and they cross over and they swap DNA. And after they've done this, they're known as recombinant chromosomes, because they've recombined their DNA. And some of the DNA from your mom and some of the DNA from your dad gets mixed up, because remember, that's what homologs are. They're genes, they're chromosomes with the same genes on them, but one's from mom and one's from dad. So during this crossing over situation, these homologs swap DNA and they become more varied. Another mechanism of variation is independent assortment. And this describes the process of the homologs separating in meiosis 1. And it just shows here that they can separate in many different combinations. So in this one, you get both homologs from your dad. And in this one, you happen to get all the chromosomes from your mom. But in this one, it looks like you got one from mom and one from dad. And this one, you got one from dad and one from mom. So they can recombine into the sex cells in many different combinations. And that's independent assortment. Another mechanism of variation, not necessarily part of genetic recombination, is mutation. So this happens during DNA replication, where things can be deleted or switched around in the DNA. And when this is non-lethal, when the organism doesn't die because of this, it passes these mistakes onto its next generation. And it contributes to some, but not very much, variation in the genes. So that's how meiosis contributes variation and allows sexually reproducing organisms to have differences in their next generations. So let's go over how meiosis and mitosis are different. It's important to note that in meiosis, the homologous chromosomes separate first. That means a chromosome from your mom goes into one cell and a chromosome from your dad goes into the other cell. But in mitosis, the sister chromatids separate. The homologous chromosomes never separate in mitosis. They all stay together. And meiosis results in non-identical cells. So those 4 cells that come out are all different. Whereas in mitosis, the 2 cells are exactly the same. They have the same genetic material as their starting cell. But in meiosis, hopefully in the end, your genetic material is all different. It's all mixed together and recombined and comes out differently. So I want you to go onto Moodle and watch the meiosis video for sure. There's another mitosis video, but the meiosis one is really important, and it explains it again in a different way to help you understand the process.